Hello, welcome to Alles auf dem Tisch, which translates to all cards on the table. My name is Bobby Rajesh Malhotra. I'm a digital artist, a self-taught bioinformatician. I was affiliated with a research center in Vienna, and my tasks were scientific visualizations and especially visualizations of protein structures in 3D. So I'm mainly also a 3D artist, and my main artistic focus points are augmented reality, virtual reality, and also a little bit of molecular dynamics, but everything not on PhD level. And I'm today here. It's really a great pleasure to have Kevin McKernan here. He's a very respected scientist, very many citations. He's multidisciplinary. And I will immediately give over to Kevin to introduce himself.
Thank you, Bobby. Appreciate being here for this. My background started in 1996 on the Human Genome Project at the Whitehead Institute for Center for Genome Research at MIT. I was working with Eric Lander at the time, and at that time I was young and just managing his research and development team for that project. So we helped scale up the DNA purification and sequencing of many plasmids out of E. coli and lots of phage as well, viruses. So we handled lots of bacteriophage and lots of bacteria at the time. I also have some experience in the biotech sector. I started a few companies, one called Agincourt Biosciences, which was eventually acquired by Beckman Coulter for its HIV detection tools and for a lot of its DNA and RNA purification tools. It was also a sequencing company at the time. And in that company, we had a company that built a DNA sequencer known as a solid sequencer that after Beckman acquired Agincourt, we spun that out, and it was acquired a year later by Applied Biosystems. And Applied Biosystems, I joined them and helped bring that platform to market. And we were competing with Illumina for a lot of the DNA sequencing, next generation sequencing market. So I've got a lot of genomics experience. I've also done clinical diagnostics work. I've spent 5 years at a company called Corrigen building clinical sequencing tests for kids with epilepsy and mito disease. So I'm very familiar with PCR and how it's used clinically. And currently we are working in the medicinal plant market, if you will. We're doing a lot of DNA sequencing of cannabis and psilocybe to understand the pathways that make these compounds that might be a little bit more open source than some of the compounds we're dealing with today. So that's my history in a nutshell.
Yeah, thank you, Kevin. So yeah, PCR is the keyword here. Me and Kevin got to know each other via the Common Drosten review report. We kind of reviewed Christian Drosten and Victor Common PCR protocol because some things quite weren't right at that time. And yeah, I asked Kevin to join the authors group, and we worked on the main review report. And afterwards, we also released an addendum that our critics won't touch at all. Because they are afraid of it or something like that. It appears like that. And the other thing is, yeah, we discussed the whole Common Drosten PCR thing in public, and it should only serve here as a hook for our main topic actually here. So just very briefly, we submitted the review report to Eurosurveillance, and we were not expecting some kind of rejection, and there were many fishy things about the PCR protocol itself technically. And do you want to add something to this, Kevin?
Well, it was certainly an eye-opener for how that journal operates. I think the lack of transparency in the review process is what's really failing. We've often been accused of not having submitted this through the peer review process itself. We put it public for anyone to critique, but I think people don't really understand that when you do an external peer review, other people's peer reviews are never put through the peer review process. If you go through the peer review of your journal, people comment on your manuscript, and those are usually kept private. And we think that's a real shame. We think the peer review process should be transparent and open to the public. So we did offer one that was public. We have not pushed it through any peer review process, mainly because we don't feel it needs it. We're referencing 20 different peer reviewed papers, and people can go to those papers and reference them and see whether those papers speak to the problems that are there. So yeah, the journal is not transparent about what happened in the review, and likewise the re-review. When we did write this this Corman-Drosten review, they did assign, they claimed, 5 other people to review what we had written. We didn't really get to see what those 5 reviewers had to say about it. One of them did volunteer some information on a podcast, Stephen Bustin, about what he thought about our report. And what's interesting is the critiques he is throwing against our report don't match his prior behavior in this field. He has been, it's noteworthy that he was on the MMR trial with Wakefield, and he crucified Wakefield for the same problems that exist in the Drosten protocol. So something's changed in his mind. Maybe the pandemic makes him think we can cheat science and cheat peer review whenever there's fear invoked, but we don't believe that. I don't believe that. I think fear is a mechanism to actually corrupt science, not to necessarily enforce it. So I think in the years coming forward, people will look back upon this in agreement with us that we should not be collapsing peer review and throwing all of the principles of science out the window in order whenever there's a virus that scares us. And that's the time to double down on it. That's the time when we need a hell of a lot more scrutiny.
Yeah, so basically, we even got the response by Eurosurveillance that maybe they even really didn't peer review it. So yeah, end of discussion of this, because it's only a hook for our main topic, and our main topic is literally in a holistic manner. We will talk about the symbol of fear since January 2020, and it's the spike protein. So I came in contact with the spike protein at my work in the research center. My task was to visualize the spike protein in open state, in closed state, in medium state, and also do some kind of slight molecular dynamics visualizations, and also locate the D614G mutation, the first real mutation, scary hype. And yeah, I quickly saw that it's not that much of a hype. Was just another next strain narrative for some research lab group. But I saw how the spike protein became the center, something like a symbol of fear, and everything was just set around the spike protein. And I was looking at it day and night as an 3D artist and also as a digital artist. And I noticed the very distinct triangle shape, and it really looked fascinating to me. And I understood why everyone kind of was concentrating on the spike protein initially as an entry point to our cells. And I would be interested into your views, just very generally, the spike protein, how you came across the spike protein, and was it the case in the past that you heard about the beta corona spikes, and there could be some kind of...
No, I had never heard of a spike protein until coronavirus, and I do agree with you. It does seem to be a very alarming word. But I think we should underscore this concept of fear, because fear creates amnesia. And people may not recall this, but I don't know if it was a D614G mutation or the alpha variant, but one of those variants was used to euthanize 17 million minks, because they were afraid if it got into the minks, it would change and then get back out and kill all of human society. And lo and behold, those same variants have since spread all over the globe and not turned into this apocalypse that they had predicted. They were hoping by euthanizing all those mink, they would stop the spread somehow, and it was all futile. But it does give you a sense of the fog of war that occurs when you start invoking things like spike and these fearful scariants and mutations. So yeah, there is a power of language going on, I think, in a lot of the propaganda out there, and it all, I think it's really embolized in this obsession over these subtle changes in the spike protein that I think we're finding again and again and again are not very significant changes. They are changes that might make an impact if you happen to have a vaccine that's narrowly targeted at spike, but they're not having a massive impact in terms of the virulence of this virus. It's getting more transmissible, but it's not getting more virulent. And that is something that is often lost in the discussion of this.
And if you look at the genetic code of the spike, is there something that is kind of distinct for you? Or in other words, there was a paradigm shift in the discussion about the origins of the
There
virus.
was, yeah, at some politically acceptable moment, I'd say.
Yeah.
The timing of that was really, really bizarre. But
yeah, the funny thing about that was we in Europe are not very much discussing about this, but you guys in the US are very much into
well,
the
yeah, we have to. It implicates one of our
we have to credit here the work of drastic research, especially they brought the paradigm shift here in the zoonotic versus lab leak discussion, and they did a lot of research or set around anonymous, not anonymous. They call them internet sloys. On Twitter, they carried together a lot of knowledge, and it was really profounded knowledge, in my opinion, especially by Billy Bostington. It's an anonymous ape head on Twitter, and he keeps shooting out.
And I think did some great work in there as well.
And the funny thing, or the fascinating thing that catched my eye, was the discussion set around the so-called furin cleavage site. It is a special site at the spike protein at the receptor binding domain, and there was some kind of a very unique insert that you couldn't find before in any predecessor of the beta coronavirus in the bats area, you know. So.
Right, that's right. Like anything that has more than 40% homology to that region, there is that we don't see furin cleavage sites in any other coronavirus like this. There's a couple areas of this spike protein. There's also the receptor binding domain, different area, but also has some really interesting amino acid residues that appear to be highly evolved, if you will, for human attachment. So if you would not expect something to go zoonotic from bats or pangolins or whatever, and then be perfectly optimized for human binding. And that's what appears to happen. If you go into this 8-sequence motif in the RBM, which is the receptor binding motif, and try to change any of those amino acid residues, you can't find something that will make it a more optimal binder. So it's already immediately in the human population and highly optimized for it. That is a smoking gun. The second issue is this furin cleavage site, which is that the virus, so in order to understand the genetic code, you have to think of it almost like the English language with different dialects. And with different dialects, you can pinpoint people from whether they're New Zealand, England, South Africa, or maybe from Boston, right? They all may use similar words, but they may pronounce them subtly differently. And that's the same thing with codons. Codons in DNA, you can use a couple different codons to express the same amino acid. And your tendency to use particular patterns like that is a real good signature for where you're from. Like if someone said something is a wicked pissa here in Boston, you would be able to pinpoint them. That's a Massachusetts Boston accent. You wouldn't hear that same phrase used in San Francisco. However, it's describing a very similar concept that might be described differently in the English language elsewhere. So these codon structures give a lot of signatures the origin of where something came from. Most of this coronavirus is very AT-rich in its codon structure, but that furin cleavage site is unique in that it has a CGG motif in there. It's GC-rich. That's not the optimal codon structure to code for those amino acids in coronaviruses. That's a signature that someone put it there. That it's unlikely to have evolved to have a non-optimal codon structure in just the one region of the genome that we don't find in any other coronaviruses. So I think that's probably the most damning evidence that there's probably lab origin here. The receptor binding motif evidence, I think, is evidence that it probably went through a lot of serial passaging and that it was already evolved by the time it made the leap into human. And I say this being someone who was extraordinarily skeptical of the lab leak hypothesis out of the gate. I think I even have some videos out there shooting down some of the earlier theories that didn't seem to be as grounded as what's come out of the Drastic group. But the Drastic group, I think, really put together a nice paper. I think it's called Sagrada et al. That goes through all of these different criteria as to why it's more likely to be lab originated than out from the wild. Now, that being said, I don't want this to scare people because lab origin, it incites all types of China phobia, and it incites all types of maybe Fauci phobia because Fauci funded it, right? It incites all types of blame games that go on, and also bioweapon concerns. But you have to realize most of these biologists were taking pieces of other coronaviruses and stitching them together, and those other coronaviruses our immune systems have some memory of. So while it sounds scary that it might be a lab leak, I think it's important everyone understand that there's a lot of evidence of pre-existing immunity to this thing or its ancestors, all right? And that we can't let go of because it's that pre-existing immunity that I think has been really important in driving this pandemic in a much more attenuated manner than people predicted. I think the fear of this maybe being a bioweapon and having this fearful spike protein name in it has driven a lot of the hysteria. And I think we have to just step back and recognize that while this may have been manipulated, most of its pieces are Legos from things that we've seen before and are capable of dealing with.
Okay, yeah. And this also brings me now to another. So this is all connected still with the spike protein. And another thing that is connected to the spike protein is I call them the spike industries. So there is some kind of a whole industry set around the spike protein. And the first thing that comes into mind is for sure the mRNA vaccine that produces the spike in open state. It's the PDB entry 6VSB, I think even. They try to imitate. And the question to you is, and you have written a blog entry on that on Substack, how you see the fail of these big pharma companies solely concentrating on the spike protein, and why don't they concentrate on all the other proteins that are attached to this virus?
Yeah, that to me is extraordinarily baffling. So for those who don't know about this, the Moderna and the Pfizer vaccine are identical amino acid sequences across the spike protein, 1,221 amino acids long, about 3,600 bases. I was somewhat shocked that they shared the exact same start and stop codons and amino acid structures in those. I just thought if two different companies wanted to clone spike, they would have been off by a couple amino acids on either end. And it would have been, and they may not have used the entire thing. One may have used spike plus something else, but there's been an incredible focus on only immunizing against the spike protein. That is a horrible idea. It's a horrible idea mainly because it is only 12% of the viral's proteome. So if you put a lot of selective pressure onto an entire population in a narrow part of this virus's genome, it's going to mutate that part of the genome so that it escapes what you're doing. And this has been a theory kicked around, but now I think the theory is maturing into the peer-reviewed literature as being fact, is that we're seeing lots of papers emerge demonstrating the selective pressure on the spike is creating spike mutations that this thing is evolving around, and the Delta variant is one of them, and Lambda as well. So these are variants that are probably man-made by putting a poor selective pressure against an entire population. This vaccine program needs to be broadened out a bit. Now, there are some other vaccines in development I'm not as familiar with. I've heard that China has one that's like a live attenuated version of this, which gives you the entire spectrum of the virus. Likewise, there are a lot of treatments now that are looking more and more favorable today because the vaccine efficiency isn't what it used to be. It was 95% they proclaimed by playing some games with statistics. We don't dig into that here, but looking at the data directly after the first shot gives you a very different vaccine efficiency than giving it 2 weeks after the second shot, and that's kind of the game that occurred in the 95% vaccine efficiency number. But nevertheless, all of these other treatments from monoclonal antibodies to dexamethasone to ivermectin to hydroxychloroquine, a whole list of them, they have better efficiencies now than the actual waning efficiency out of the vaccines against the Delta variant, which is making a lot of people consider, all right, we need to get the treatment thing under control. We need to know how to use these things because regardless of whether you're vaccinated or not, you're probably going to need to know about treatment because many people who are vaccinated are getting hit with this. The benefit of the treatment side is that your immune system builds immunity across the entire 29 kb of the virus. That is more likely to be durable and be remembered and last a lifetime. It's only these folks who are getting this narrow immunization against the spike protein where we're seeing the vaccine evade it, or the virus evade that pretty quickly. So I think we focused too much on one area. We put all eggs in one basket on the vaccine program, and that has led us into the mess that we have today.
Mm-hmm. Exactly, and we are kind of in an endless, I call it a circle jerk. We have a spike mutation.
Booster gerber wheel.
We need a booster, and then we have another spike mutation, and then maybe a whole different kind of spike protein of another virus comes along. Or it's just, it's...
The boosters aren't even different. The boosters are the same spike. So it's like it's pouring more of the same antibiotics onto your antibiotic resistant bacteria. So no effect.
This should be our closing words. The spike protein as spike industries and also as a symbol of fear. We should end this in this case, focusing on the spike protein. We should get a more holistic approach on pathogens and how to solve the problem here. And as you told before, just focusing on the spike protein as a mRNA vaccine, which is actually great tech, without a doubt.
Absolutely.
It's the future. It's the future of Western school medicine, no doubt, and it's also the only hope for big pharma to kind of avoid expensive bioreactor production and just let the human body produce via your own ribosomes. So
that's very true. It's a good point you brought it up, that there's a lot of promise in the technology. This is probably its worst inning. Right, the first time we've ever used this in grand scale, they're bound to make mistakes. I think the spike protein is, I think retrospectively, we're going to look at this as the wrong place to showcase this technology, because we chose the most toxic part of the virus's genome to express. Looking back on this, we probably should have done this very differently and chose a bunch of other regions of the genome to target, as opposed to the one that the Salk and the Scripps have shown is a very, very toxic peptide. Now, I want to caution the toxicity papers. I encourage people to go look at them. These are all done in vivo. I don't think we've seen direct hard evidence of this being prion-like. There is toxicity mentioned that this thing has a lot of characteristics of prions based on a bioinformatics perspective, but there's certainly adverse events that are piling up that mimic some of the concerns that people would have over some type of spike protein being toxic. Other parts of the virus would be better places to target these tools. The second issue about these tools is the delivery mechanism is right now ubiquitous. It transfects any cell. The virus doesn't do that. As you know, the receptor binding domain brings this thing into ACE receptors. And there are some other receptors it hits too, like CD147. But that means that your cells, when exposed to the virus, are getting a very different picture than when exposed to vaccines. Maybe in the future, they'll target these. They'll make them so that the targeting is a little bit more in line with what the actual virus does. If there's a way to make these target ACE2 receptors like the virus, we might get a very similar adverse response profile with the vaccine that we get with the actual virus. But right now, I think there's some divergence there. So anyway, yeah, the messenger RNA technology is very exciting, but this is its first inning, and I think you're going to see a couple strikeouts in the first inning.
Thank you very much, Kevin, for your very precious insights, and we will see you soon, hopefully.
Indeed. All right. Thank you for the time, Bobby.